Thank you all for coming. It is really my pleasure that I can communicate something for you today. And I would like to say that I know that the ruble is more interesting, but let's just give it a try and get back to science. What I'll be talking about today is something that I really am interested in, and I've been interested in my career. It's about medicinal chemistry, or the drug design, or how scientists design drugs to treat diseases. The disease that I'm interested in is cancer, and what I'm also interested in is how chemists and biologists and scientists actually use natural products to design therapeutic compounds that can help in curing cancer. So this is what my talk is going to be about. It's about how marine organisms, so natural products from the sea and from the oceans, have helped in combating cancer. First of all, this is just a quick definition of the science that is called pharmacognosy. It is concerned with active substances that are studied. And the objective of the science is to find and develop new therapeutic agents from nature. Now, before I start, just a little bit of history. I lost orientation in that wheel. So, before 384, 322 before Christ, the Phoenicians were the first ones to start exploring the sea and exploring what's in it and trying to find out how it works. But unfortunately, it wasn't recorded, and their works were not really referred. The first real person to record some information was Aristotle, and he was the first person to observe differences in organisms in the sea and to actually note them down. The second one was Captain Cook. He is well known because he traveled a lot during the 1700s. He actually made around the world twice, and the discoveries he has made were actually very important in understanding how marine organisms and how the marine life actually works. The third one, which is someone that you all know probably because of the theory of evolution, is Darwin. And Darwin's theory of evolution has actually originated from his love to the marine life and his interest in understanding how different organisms have developed, how different organisms shared things in common, which helped him formulate the theory of the survival of the fittest and all the theory of evolutions that you know about. Now, why are marine compounds, why are they helpful in drug discovery? First of all, just a little bit of information. 10,000 compounds have been isolated until now from the sea, and they have helped in combating a lot of diseases, including cancer. The second thing is that those compounds have been isolated from a lot of different organisms. The tunicates, the sponges, the algae, and even the animals. So there's a lot of organisms right down in the sea that can produce a lot of diverse compounds that can be really useful in understanding how to combat disease, especially cancer. Now this is an important pipeline for you to understand that if we start with isolating compounds from the sea, we can have up to 10,000 compounds, but in the end, after studies and after identification, we don't really end up with much to give to the market. We end up with just 7. So we really need to isolate as much compounds as possible to increase the chance of actually ending up with an FDA-approved drug that we can actually use to treat cancer patients. Now, why marine compounds? The first thing about the marine life is its richness. There is 33 human phyla, if you've heard about phyla. 11 of them are only terrestrial. 28 are in the sea. And therefore, the richness of what's under the ocean, what's beyond what we don't know, is actually very, very, very rich. And this is why it really pushes us to understand what is really down there and how it could really help us. It really took us ages to get out of the water, but there are a lot of other organisms, much more than us, that are still living there. And understanding how they live and identifying the compounds they use to live is actually very interesting in understanding how we live as well. The second is the biodiversity, as I've just mentioned. There's a lot of biodiversity under the sea, much, much more than what's on the earth. The difference in plants, in algae, in mollusks, in starfish, and in animals, and in mammals, creates a very big pool of things that we can use to study. The third and most important thing is the inspiration from self-defense. You see, 3,500 million years ago, organisms started to develop in the sea. Now, for them to be able to stay alive until now, they had to undergo a lot of competition, because living in the sea is not really that easy. You always have predators. You always have the pressure, the salinity, the pH. There's a lot of chemical and physical environments that the marine organisms have to deal with. Now, some marine organisms have physical abilities to deal with them, sharks, but some others are really slackers. They just stand there like a starfish, but they don't have anything to do. So they produce chemicals. This is how they defend themselves. And so, if you think that we were the first persons to use chemical weapons, then think again, because these jellyfish have done it long time ago. So we learned from them, but I'll tell you how we learned from them as well. The fourth and and also important thing is potency. Potency as in the power of the compounds that marine organisms use. The power is really high, because if you really think about it, for a compound, a small chemical compound, to work in such a huge sea, it has to take into consideration the salt, the salinity factor, because it is very, very, very diluted. So the compound has to be very potent in order to work. And so all of these together led scientists to start exploring the sea and to find out about natural compounds to combat diseases. And the discoveries were really, really, really a lot. I repeat, it was like up to 10,000 compounds, from invertebrates such as jellyfish, from vertebrates such as the fish and the sharks and all the animals that we're familiar with, and the microorganisms as well. Bacteria, fungi, cyanobacteria. And so now you may think that this is all very interesting, but how does it really work? And honestly, I really didn't want to go into too much details, because sometimes I don't even understand them myself. So, but I'll try to show you how we can really get from this into this. And honestly, it is not that hard. So it all starts with an expedition. We all go to the sea for 1 year or 2 years, and we try to get as much information as possible. We send them for isolation, we ferment them to try and get some more of what we already have. Then we try to extract them to get them pure, and then we start screening. And what I mean by screening is that we start to check whether or not they have activity in a human being. And one of the most amazing examples that I have found in screening is the National Cancer Institute, what it calls the 60 panel. So the 60 panel, every year, the National Cancer Institute welcomes a lot of compounds from an expedition, and it runs them on 60 cancer cell lines to check whether or not there is activity against one cancer cell line. And then when there is activity, it takes them to bioassays, and then they try to sort of optimize this compound that they discovered, and to end up, hopefully, with a clinical compound that can enter the clinical market. So, how do we do this again? We can either wade, wading, scuba diving, and we have dogs that help us for that. We have the nets, and these are very efficient as well. Then come the biosensors, and here the budget increases a lot. And then the submersibles, of course. Now, if you want to take into account one day for that submersible to do an amazing job, which can lead to the discovery of one compound in just 2 weeks, it really costs up to 45,000 euros per day. So it really needs a lot of technology so that we can end up with discoveries in a short time. Now, the final thing that I want to get you through is the evidence. Which compounds have made it, actually? And I will show you the compounds that have been FDA approved, which are used now, and which have been used to save a lot of lives, especially children, because they get cured easily. The first one is brentuximab, which is known as the Adcentris in the market. That's the market name, and it's taken from this mollusk. And the interesting thing is that this mollusk produces something such as this, which has helped the scientists produce an antibody to sort of track a drug, an anti-cancer drug, into the cells. Before that antibody, this drug produced a lot of toxic effects for patients that were being treated with cancer. But after this antibody has been selectively anchored to that drug, then the treatment efficiency improved by accidentally discovered. It makes this nucleotide, but the only difference between that nucleotide and the nucleotide in our DNAs as humans is this sugar. The sugar is different. But what did scientists do? They said, let's just try it and see whether it can act like a Trojan horse to stop the DNA of the cancer cell from multiplying. And it worked. And it worked. This selectively binds to the DNA of the cancer cell, which is multiplying, and it inhibits, it stops that DNA from going further, and therefore it inhibits cancer. Another example is this, this tunicate right here, which also made a lovely compound. I see it as very beautiful. So, I mean, nature provided us with that, and apparently when we tested it on humans, it stopped cancer from multiplying. Another example, and the last one, is also this amazing creature right here, which also made that molecule that we only had to tweak with it a bit to make it optimized for treating breast cancer. So, these are the compounds that are currently undergoing research, and they've entered clinical trials. And as you can see, they come from different organisms: tunicate, worms, fungus, mollusk, bacteria, and they treat a lot of diseases. The ones I've selected is cancer because I just wanted to narrow down this talk. So, in conclusion, the thing that really makes me interested about that talk is not that I work with it. It's not really my project, but I love chemistry and I love nature, so I just try to combine them together. My idea is poison kills poison. And we are very different in terms of species. But if you think about it, the jellyfish and I share the same atoms and the same molecules. So if I were to ask this starfish or this jellyfish or that sponge, how did you do to combat yourself from infection? How did you do to combat yourself from pressure? It can give me answers. So with that, I'd like to thank you all and keep the oceans clean.
Thank you very, very much for a lovely talk. Questions? Please.
Thank you very much for a lovely talk. It was really, really interesting.
Thank you.
It was really lovely to see a fellow naturopath here.
Thank you. I'm really happy. It's just too cold. Cool.
It's really normal. But my question is about pharmacognosy and marine pharmacognosy. So if you compare plant drugs, the drugs derived from plants, like not marine plants, they're much more numerous rather than marine compounds derived from the sea. Why do you reckon it happens? Is it because of the lack of research, or is it because of cost effectiveness? Because as you said, it was really expensive to get the compounds from there.
Plants are actually much more rich in compounds, and that is true. I think it's not because of either. I think it's just because of what I talked about, the physical and chemical ways of self-defense. Plants are one of the least that have a physical way of self-defense, and this is why compensation starts being on the chemical side. They can also produce a lot of compounds, maybe better than evolved mammals or evolved mollusks, or sometimes those other organisms can move. They can capture. They can inject ink. They can do a lot of stuff that can be maybe easier to defend themselves with. So I think that's why.
Если у кого-то есть вопрос, вы стесняетесь задать его по-английски, я могу перевести. Пожалуйста. Okay, I have a question.
Which one? There's a lot.
Both of them. The one in the middle.
There's a lot. Like 1, 2, 3, 4, 5, 6, 7, 8, 9.
Okay, so was any of that used to treat any diseases?
Yes, except the mermaid. I think the mermaid was used to treat other diseases. Anyone else? No? Oh, that's a big question. That's a big question.
Is it a failure or a shortage?
I think the problem is not in these compounds. The question is a bit bigger. The question is, why does cancer therapy fail? And there's a lot of answers for that. There's a lot of factors. The most important of which is that we are different. We are very different, and we can react to the same drug, even if it was Panadol or even if it was Advil, very differently. I can react to it different than you, and it's the same thing with cancer. Also, it depends on the staging of cancer and the type of cancer, and whether that cancer is actually resistant to tell you, no, I don't want to be treated, or to tell you, I'm very weak, you're killing me with that starfish. So it really, really depends on a lot of stuff. However, natural compounds have been very, very useful, and the evidence is that 50% of the anti-cancer agents that we now use are derived from natural sources, which means that my point is, as long as you don't produce a lot of synthetic things, it has more chance to work.